Okay, welcome back, everyone. I'm very excited to kick off our first keynote presentation of the morning. The presentation will be by Dr. Tychele Turner. She's an assistant professor of genetics at Washington University School of Medicine in St. Louis. Her lab focuses on precision genomics of neurodevelopmental disorders, especially autism spectrum disorders, with a focus on de novo and rare inherited variants, the potential underpinnings of sex biases, birth order effects, and clustering of disease mutations in primary protein structure. Dr. Turner earned her PhD in human genetics and molecular biology from the Johns Hopkins University School of Medicine in 2014. She then served as a senior research fellow at the University of Washington Department of Genome Sciences from 2014 to 2019 before starting her lab at WashU in 2019. A major area of focus in her current research is in identifying and understanding variation in the non-coding genome. And for this, she uses state-of-the-art sequencing approaches, including both whole genome and whole exome sequencing, and using both short and long read sequencing. After variant discovery, a major research effort is then into variant interpretation and prioritization, and uses a combination of functional genomics data, massively parallel reporter assays, other functional experiments using to understand and assess the consequences of variation. Of particular note to this meeting, Tychele is very innovative in her computational methods and platforms used. Earlier this year, her lab published an algorithm called horse and hare that I think we're going to hear, the hare and the tortoise, I think we're going to hear a bit about. For de novo variant detection. So de novo variants play a major role in autism spectrum disorders and contribute to maybe, I don't know, half or so of sporadic cases. So what was really clever and impressive about the computational work was there's kind of a couple of versions of the algorithm. The slow version, the tortoise, uses regular CPUs, but the fast version, the hare, uses GPUs and cloud computing, including on the AnVIL, to be able to analyze tens of thousands of genomes to better understand the genetics of autism and other major disorders. It's my great pleasure to invite Tychele up to the platform today.
Thank you so much for the introduction and for the invitation to come speak at the first AnVIL meeting. It's very exciting to be here today and to talk about some of the work in my lab. So I'm really trying to focus on the parts of the lab where we've used cloud computing. And I titled my talk, Unlocking the Power of Precision Genomics in the Cloud, because we mainly focus on a topic called precision genomics. So in my lab, we're utilizing genomics for rare and common disease. And in particular, if you think of the kind of workflow that we have, we're going to have an individual presenting in a research study, in the case of my lab, or in the clinic in a more general sense. And once that participant is enrolled, then we're going to do some kind of a biospecimen collection. We're going to do some kind of sequencing. You may do something else, but primarily my lab does, sequencing. And then bioinformatics, computational biology, and function are all lumped together in this big category that goes into variance and interpretation. And in my lab, we're actually interested in all aspects of this workflow. And so we're trying to find all the points that we can improve in those. And mainly we focus on autism and other neurodevelopmental disorders. So when I opened my independent lab in 2019, we started publishing papers, and as we were publishing these papers, I was throwing around a term called precision genomics. And the reviewers of the papers were not too happy. They actually said, what is that? What is the definition of precision genomics? And if there isn't one, you should define it. And I was like, oh yeah, we're going to define it. So we defined it as determining all possible relevant genomic variation within an individual to the precise nucleotide. And a lot of times when I give talks, people say, oh yeah, that's precision medicine. And I say, no. If precision genomics is all that precision medicine is, we're in big trouble, right? So precision medicine should be a much broader angle than just the genomics. But the precision genomics was very much inspired by the precision medicine initiative, which was brought about by the Obama era a while ago now. But it was the idea of individualized care that's tailored to you. And what I love about it is that it brings the patients and the providers and the researchers together. So for once, there's a place for me, a PhD. I can be involved in the process as well. And so precision genomics is just one area under the umbrella of precision medicine. And so I started to think about, what would it look like if we actually got to precision genomics, if we knew every relevant variant in every person down to the very precise nucleotide. And so I started to think about, what do we know? So instead of like, what don't we know, what do we know? And so what I think is really intriguing is that we can think about the advances we've made over the years, and we've made incredible advances to the point where if we have a single variant, and I have this large bar is just one large variant, large effect variant, and this is representing something like an autosomal dominant loss of function in a syndrome, right? So if you can find 10 people in the world who have that syndrome and it's due to one gene, you're going to find the gene. Like, we're really good at that right now. And I think that, that's been a huge improvement. On the other side of things, where one of the sessions is about even today, is polygenic risk scores. So now we have many, many little variants. So hopefully you can see there's tiny little ticks there, and they're additively adding up to this. Well, it doesn't have to be additive, but essentially they're going to the level of the large effect variant, let's say. So they're causing the phenotype. And what's really interesting about that is we've gotten really good at predicting these risk scores, and people, we struggle with diversity and ancestry and representation, which is super, super important in terms of polygenic risk scores. But the fact is, we do know how to calculate them. So we do know how to think about that end of the spectrum. So we have one end and we have the other end. And often in the past, we had the rare variant people on the far left, and we had the common variant people on the far right. And sort of we can wave at each other across what I would call the valley of the unknown, right? So everything in between there is still really hard for us to do. And so I think what we need to do is to actually build that bridge of discovery where it's 2 variants, 3 variants, 4 variants, 5 variants, etc., all the way till we can get to the side of the polygenic risk scores. And it seems like that could be easy, right? No, actually, it's really hard because the valley of the unknown is littered with other complications. So not only do we have our DNA, right? We've got our environmental exposures like the sun, the food we eat. We've got viruses, right? COVID was a perfect example, but other viruses like EBV that can integrate in our genome. We've got not just the variant itself, but where it sits within the gene regulatory network. So every gene is not equal in our genome. And so this big valley of the unknown is sort of what we have to bridge. And I'm not quite sure how we're going to do it, but we're going to do it starting at least, I think, step by step. I don't know what the final picture will look like, but I'm hoping all of you will be interested in working on this as well, and I think you are. So as I started my lab, then I'm thinking about what we know, what we don't know, and I decided, okay, 10 years, the first 10 years of my lab, we're going to solve these 5 problems. Not in isolation, but we're going to make great progress, and it's going to be very exciting. And I'm 5 years in, and I'm thinking, oh wow, that was a little ambitious for 10 years. But these are the areas that we're really interested in. So the first is non-coding variation. We know that the bulk of signal, for example, in GWAS is in non-coding regions, and yet we're not super well prepared to, say, when I see a non-coding variant, what does that mean in an individual? And so there's a lot of work to do there. There's the variants due to genomic technology. So the standard short read sequencing in the clinic is missing variation, and a lot of the work we do in this space is in long read sequencing. And then I was thinking about, if we had precision genomics, it would look something like this. And mind you, we have to consider there are lots of ethical considerations. I'm not a scientist who does ethics research, but that's something that's super important. Although I am ethical in my research, I just not in that area of focus. But what I was imagining was that one day I'll be able to go to my doctor, and at the beginning of the visit, he'll collect my biospecimen, he or she, and by the end of my visit, they'll have my genome in hand and be able to do something useful. So I was thinking a lot about, oh man, if that has to be like in an hour, we've got to speed things up. And I thought the easiest way is to speed up the computation. And so we're very interested in that part. The next area is a combination of rare and common variants. And I don't mean across the bulk cohort, because we're all pretty good at doing that. But I mean at the level, if I have a person standing in front of me, how do all the common and rare variants in their genome result in the person standing before me? And then, of course, we're not just a product of that genome, we're a product of that genome and interacting with the environment. So that's the last area. And so here's the progress we've made so far. You can see it's a couple of the papers are in 2 categories, but we're sort of working towards those fourth and fifth areas of research. But today I'm mainly going to focus on the speed of the current genomic workflow because I thought that would be kind of the appropriate topic in terms of the AnVIL meeting. So along with these papers we've produced, we've generated lots of software. And a lot of people say like, oh, you're just a tool builder. And I'm like, well, no, like we're building the tools so we can apply them to neurodevelopmental disorders. But as we're building the tools, we might as well make them available so other people can use them as well. And so that's kind of our main focus is like, we want to solve interesting problems in relationship to neurodevelopmental disorders, but like we want to have highly reproducible work, and AnVIL and Terra are really important in that process. So I'll just briefly introduce you to the software, and then I'm going to talk about two of them today. So the first is HAT, which is hare and tortoise, which I'll talk about, which is trying to find de novo variants in short read genomes, long read genomes, and short read exomes. And then we realized that QCing these variants, actually, you have to, we were writing codes over and over. So we just made a R package called Acorn that you can quickly do QCs. We have a database called DenovoDB, which we just released the newest version on October 8th, which was my birthday. And there are now more than 70,000 parent-child sequence trios from the published literature and greater than 1 million de novo variants represented there, and it's freely available. We also developed a tool called 3D Clump, which looks at clustering of missense variants in 3D structures, 3D protein structures. And then ACEs was the tool we actually developed and published on when we had our AC2 cloud grant. And I won't talk about that one today, but that was our first venture into the AnVIL. And then I will talk about CNPI, which is a new tool, and I'm hoping you may be able to give some feedback. And then Diamonds is also coming soon, but I will not be talking about that today. Okay, so the first tool is called Hare and Tortoise, or HAT, and it was developed by Jeffrey Ng, who's in my lab, and he'll be here at the meeting Wednesday through Saturday, and he'll be presenting a poster actually on that 3D clump, but he'll be here around if you want to chat with him at all. And so what is the first issue, right? So the issue was we wanted to generate a caller for finding de novo variants. And so a de novo variant is a variant that is in the child, but not in the parents. So all of you in the room have 40 to 100 of these in your genome that aren't in your parents' genomes. And, most of the time they don't really do anything, right? We all have them. But in some cases, when they hit important parts of the genome, like protein coding regions, they have been identified as enriched in many phenotypes, like neurodevelopmental disorders that I study. And the main research generally is on disease relationship. You'll see this also in like Mendelian genetics projects. De novos are coming up a lot. Or mutation rates, if you're interested in that. So what happened was when I opened my lab, I was coming from Evan Eichler's lab, and I had developed a de novo caller, and that caller worked in about 800 hours, CPU hours, from cram to final product, and it incorporated multiple callers at the level of the cram to the VCF. And that's not great. That's a long time. But we didn't have a lot of time. I didn't have a lot of time in my postdoc to try to optimize it. But one of the first things that happened was my R00 was on non-coding. And I'm like, oh my gosh, we're going to have to analyze lots and lots of genomes. And we have to accelerate this. So we thought about different ways to accelerate our code. And in the end, we just developed a brand new tool. It actually isn't the same tool that I developed before, but that's okay because we found improvements and we found ways to make it better. But we thought about how we could write better code. So one is we could just write it in a different language, Python to C++ or Rust, which is gaining a lot of popularity. We could parallelize, which is an obvious thing to do, or we could utilize GPUs. And we ended up utilizing GPUs because some of the people at my university were like, NVIDIA is super interested in getting into genomics, and they're looking for people to work with. Like, are you interested? And I was like, yeah, of course, this will be amazing. So just to give you a quick background, although knowing all the people here now and having discussions at different meals, I think that, you probably already all know the difference between a CPU and a GPU. But in case you don't, a CPU is very much, your standard workhorse. It's on your laptop. If you're here today and you're watching a video while you're also listening to my talk and working on a grant, all these kind of things are really useful. Like CPUs can do it all. But then the GPUs came online, and one of the first advances were in developing video games, which is super cool. So we've gotten the video gamers to go all the way to genomics. I think that's an incredible advance. But basically, they have lots of cores, and if you know how to program on them, you can parallelize things really, really fast. And so what we did was we did just that. We incorporated a tool called NVIDIA Parabricks, which is a super fast GPU accelerated caller for GATK and deep variant. And then we fed those into a program called GLNexus, which is quite popular for joint genotyping. And then once we had all that information, so we have like two trio level VCFs, then we fed them into our de novo workflow. And what happened was by the time we got done optimizing everything, we could run this in about 8.5 hours, and in less than an hour with further parallelization. So from 800 to 8.5, and I was like, oh my gosh, this is amazing. I love it. And I should say for exomes, it's about 7 minutes to sample, so it's even faster if you have exome data. And then what we found was that, with exomes, I'll show a little bit later on, we had to add a little bit more filtering. But let me tell you about this first paper. So we were very enthusiastic that we had a fast tool, right? Like, that's great. But what if the tool is not good? Okay, so something could be really fast but not work correctly. And so what we did was we used this monozygotic twin pair, and what we asked was, how often do we find the same de novo variants within those two genomes, right? Because they should be essentially the same. And we found there was a high level of concordance, so about 75. And if you don't work with de novo variants, like, you may not gain how difficult it is to actually find de novo variants. It's really hard. Like, I told you 40 to 100, but most people, their first implementation of a de novo variant caller, they come up with like 1,000 variants. So getting to 40 to 100 is actually really tricky and is a really hard problem. So we thought this was very encouraging, though, that our tool with the GPUs and everything is getting to 75. And we also know that about 20% of all de novo variants occur at CPG sites. And so that was good. This was about right. So we were like thinking, what are we going to apply it to? The first thing we thought of was 1,000 Genomes Project, because the 1,000 Genomes Project released data on 602 trios, and they are up in Terra. And that's great. It's a public data set. And so we could also, share these de novo calls with other people. So you can run HAT yourself, right? And hopefully you can get the same results as us. And this data represented a lot of diversity. So we have, and these labels all come from 1,000 Genomes themselves. We didn't make the labels, but they have admixed American, African, East Asian, European, and South Asian. And so we just went ahead and ran it. And okay, so the QCs that I'm showing here are now available in Acorn, just as a side note. But here's what we saw. We actually had the 602 trios, and we had about 10 times more de novo variants than we were expecting. And this actually surprised us. And then we thought, oh no, HAT's broken. It doesn't work. It worked on one trio, one family. But then we turned to our autism data sets from the Simon Simplex collection and from Spark. And so these are 4,216 trios and 1,326 trios. And we saw that the number of de novo variants was about 75. So we were very curious what this could mean. So we further looked at more QCs. So we looked at the CPG sites, and we saw that in these two cohorts, it was about 20%, and in 1,000 genomes, it was less. And then we looked at the percent phase to the paternal chromosome. So even though these are de novo in the kid, right, they're arising on one of the chromosomes, either from mom or dad, particularly in the germ cells. And so we saw that we should expect them to be at 75%, which we saw in the two autism cohorts, but not in the 1,000 genomes. It was lower. And actually, as the number of de novo variants increased, this dropped down to 50% about random. So we were very curious what this could mean. And we noticed that the DNA came from different cell types. So for our 1,000 Genomes Project, the at that time, but there's been a lot of incredible advances since then. They're doing a lot for pangenome, they're doing all blood and everything. But at that time, they were using LCLs. And so these are cells which you get blood, and then you use EBV to transform them, and then you grow them in the lab. But what happens is like maybe you can get mutations during that process. And we thought maybe that's what's happening because when we get from blood and saliva, we don't see that kind of difference. So we took a look at the different populations and we saw that the oldest population, that oldest, not like ancestrally, but meaning the one that's been cultured the most, had the highest number of de novo variants. And we thought, well, that could indicate that these are not all de novo variants, but rather just like cell line variants that are occurring over the culturing process. So we had some people at the McDonald Genome Institute at WashU, and then we also went to the literature and we were able to track down different samples over time for a sample called NE12878. She's the most sequenced person in the world. She probably doesn't know it. She's somewhere in Utah, I think, if she's still alive. I don't know that, but she's been sequenced like every time a lab has to test something, they run her sample. Well, it turned out we could actually take the variants that we found with HAT, and we could actually like build a synthetic, kind of small synthetic genome out of that, and we could build a tree based on what variant was present in that data. So like we just converted them to IUPAC codes and stuff. And what we saw was that the sample that had been sequenced in 2012 was super far from the sequence in 2020. And of course, they're all really far from the reference because these are representing sites of potential de novo variation. And you can see that there are lots of sites that are shared, so 1820 that are shared across all the different replicates, but then there are differing levels of sharedness. And down below are the percent that are actually scoring as correct. We did a lot of secondary analysis to try to make sure that it was all correct variants and everything. And so we thought perhaps then this is lending evidence towards these cell line artifacts. And then we took advantage of something that's happening in the cancer world, and that's this idea of generating mutation signatures. So mutation signatures are super cool, right? So you can basically take the patterns of mutations that you see in a given genome or in a given set of regions, and you can ask, do those mutations look like a certain signature for different types of cells or types of processes? So what are called the signature one are age-related patterns. And those are the types of patterns that de novo variants look like. Because if you can imagine how the biology works, right, that typically most of them are coming from the male germline, which is continuously replicating over the life course. So it's like an age-related pattern. But what we saw was that as we looked at people who had lots of de novo variants, they were falling into these signatures of B-cell lymphomas. And I didn't mention it, but the types of cells that are transformed by EBV are B cells. So in fact, by immortalizing the cells so they can grow really well in the lab, which is super great because you have them forever, you've actually turned them into like a cancerous type of cell, which is okay. Because if you're not studying de novo variants, it doesn't matter. Like it literally doesn't matter. But if you're studying de novo variants, this can be tough. And so we started to look at what the variants were affecting, and we saw that some of them were affecting DNA repair genes. And so these are genes which, if they have variants in them, could result in more mutation. We also borrowed some tools from our autism research, which is looking at the enrichment of de novo variants per gene. So you can use information like the human chimpanzee difference or the triplet sequence context in the genome to predict how often you'd see a de novo variant in a given region. And we found 29 genes shown here that were enriched, but only one that was significantly different between the people who had the correct number of DNVs and the incorrect number of DNVs. And that was called IGLL5, which is also important in B-cell lymphoma. So we did not mean to go down this path at all. But sometimes when you're building a tool and QCing it, you run into these kind of things. And so we also identified some variants that clinically would have been recognized as pathogenic. And so we just put like a little asterisk that if you're doing de novo variants on the original data set, you might want to think about like which particular set of variants. And we made all these variants available to the community so people can, check that out. Okay, so what happens then is that we got a grant from someone else, and they said, we don't care about non-coding. We want you to do exome analysis. And when I left my postdoc lab, I was like, I'm never going back to exomes. You should never say never, because, they're like, we've got 100,000 exomes. Yay. And so, which was super cool. Like, I'm not downing it. It's great data set, and we love the data set. But it turns out when we applied HAT right out of the box, we found that there were differences in the de novo performance in the capture region versus sort of outside of it. So when you capture an exome, right, you're using little probes, and they're trying to bind to sites that are in the exome. But sometimes, like, the reads are actually hanging over the edge outside of that capture, and you sequence those a little bit, but not to as high a coverage as the actual capture. And so we added a filter to HAT, which actually says if your variants are within the capture region plus minus 10 base pairs, then our confidence is that there's about a 92% confirmation rate. And those are going to be really good. If you're going to the outside of that, there's going to be low confidence. You should do something else just to make sure they're real. But that's the one filter we had to do. Well, then we got into long read space. And these were two papers that I'm not going to talk about today, but were pretty interesting, where we solved a family with autism that had been around the world that had no answers. We did long read sequencing, and we found the answer. It was super, super fun. But what happened was we had that data sitting in our lab, and then we also study 9p deletion syndrome and 9p duplication syndrome, but we had sequenced another family with long reads on 9p. And so we thought, let's take all of this data and apply it and see if we can make calls with HAT as well. And what we found was that there was an interesting observation. So a while ago, there was a big argument in the field. Maybe it was behind closed doors, maybe it was out in the open, but it was like, do we want a 22x genome or do we want a 30x genome for short reads? And I think, like, a lot of people fell on the side of 22x for general purposes, and then there were some people who were saying 30x for de novo variants. And that turned out to be the most helpful. So what we did was, well, we did a lot of things, but I'm only going to show one particular slide, is we took this family, which was originally at 46x for the kid and about 30x for the parents, and we downsampled to 30x, 20x, and 10x. And then we looked at the number of de novo variants that we found. And the first number is genome-wide, and the second number is in unique regions of the genome. And that's the kind of part of the genome that short reads would perform the best in. And long reads actually just perform well everywhere. And I don't have a lot of long read information to present today, but I think Benedict is presenting on long reads tomorrow, so that'll be great. But long reads are really the best, if I had to say. But anyway, they're awesome. But what you can see there is the number of de novo variants and then the percent that are identified as correct. So you can see that at 10x, we find 93 at a 62% confirmation rate. At 20x, we find 153 at an 87% confirmation rate. And kind of the sweet spot really was at 30x. So we found 123 overall and 82, which is in between that 40 to 100 in the unique space, with about a 92% confirmation rate. And then once you got out a little bit further, probably because the parents are really only at about 30x, there was no substantial gain. So one thing that we thought was really cool was that pretty much out of the box, HAT worked on long reads. So if you are doing long read analysis, you can use our tool. If you're doing exome, if you're doing short reads, it's all there. And we also have it available to run in Terra. And so I just put a few screenshots in. So this is our doc store for hair. You definitely want to run hair with GPUs, and you'll see why in a minute if you're going to be running HAT in the cloud. And just remember that we're starting with CRAM, so it's a lot of analysis steps, which is why when you see the price point, you'll understand. And here's the workflow. So this is the WIDO and that came in from our doc store. And we love doc store and we love WIDO, so it's really fun. Here's the empty input JSON, and here's setting up the hair run. And then this is the job manager afterwards. It's a tiny bit blurry, but there's the different steps you can see, like the GL Nexus DV, GLX HC, that's haplotype color or deep variant. And then here's like the Google billing cost table at the end. So here's the current average cloud cost. So I put all of them for reference. So I'm actually partitioning the parts. So here's, if you run Parabricks deep variant and assuming all of these different parameters, you can run a sample through it deep variant for about $1.14, at least in our hands. And that's pretty cheap. If you run deep variant out of the box from Google, it's about $11. So we definitely like the Parabricks version. And I don't have any affiliation with any of those companies. I don't have any consulting. I don't get paid by any companies, just so you know. So this is just our unbiased observations. And the Parabricks with GATK is about 67 cents and regular GATK is 17. So if you run hair, it's about $9 a trio going from CRAM to DNVs. And remember, you get the trio level VCFs on the way. If you run Tortoise, which is all CPU based, $114. But the reason why we have Tortoise is because the reviewers said it's not fair that you have a tool that people who don't have GPUs can't run. Like, if they don't have GPUs, they can't run your tool. And well, I guess we should have just said, go to Terra and you'll always have GPUs. But at the time we were like, all right. So we made Tortoise and we made sure it was functionally equivalent. If you really want to run Tortoise, you can. Don't recommend it, but it actually let us have the cool, I guess, logo with the hare and tortoise. So in conclusion for this part, we utilize these GPUs to develop this very fast, accurate de novo caller. It's all available and can be run in the cloud. And we ran into a little bit of interesting biology along the way, and now we're applying it much more broadly to autism genomes. So the next tool I wanted to talk about is called CMPI. It doesn't have a logo that we've agreed on yet in my lab, so it's just a big yellow box. We actually have a logo, but we haven't come down to if that's the logo we really want. But this is a tool that was developed by Jack Ustanik, who's pretty new to my lab, and I've not presented this anywhere. It's not published, so any feedback would be appreciated as we're going towards the process of publication. So the second area that we're thinking about, right, not just these small SNPs and indels, but in looking at copy number across the genome. And there are lots of ways you can call copy number and structural variants. There's like a million. I just did a query of GitHub, and there's hundreds, if not thousands, of different tools. But one of the things we're really interested in my lab is looking at non-coding regions of the genome across like millions of genomes, and being able to say something about those non-coding regions. Like, are they constrained in terms of dosage across lots of samples? And so we started looking at different tools, and the tools we looked at were from the Kid Lab, which is FastCN, which is a multi-mapper. So these are read depth-based tools. I'm just going to be talking about read depth for today. And the other one was a K-mer-based. We actually tried to develop our own, and we actually liked Jeff Kidd's a lot better, so we're going to use that one. But the biggest difference is between paralog specific copy number and not. So here I'm showing you data from a family with autism, and it's a trio. And this is their estimated copy number over these tricky genes in the genome on chromosome 22, CYP2D6, CYP2D7, CYP2D8P, right? They can be confused with each other. And in fact, when you do multi-mapping, you let the reads place wherever they map, right? You can put them multiple places. And you can see our copy number from that is very high, like 4 or 6. But when we use the K-mer-based estimate, we actually get this beautiful copy number of 2 in the mom and the kid, and we get the duplication we expect over this gene in the dad. And so we have decided that this is going to be our strategy to look at read depth-based copy number across lots and lots of genomes. And so we decided, first of all, let's make sure we can do it in the cloud, because we're running this on CCDG, we're running this on other data sets in Terra. How can we make it really cheap? And how can we make it work well? So basically, I should quickly say that QuickMer2 output is essentially copy number in windows, every window across the genome. And on average, there are about 1,000 base pairs. So we want to have that little small megabyte file instead of a large CRAM. And so we tested it out. We use these different parameters using different memory drives, no GPUs in this case, and then whether we could tolerate the use of spot VMs. So these are these preemptables where essentially if you put it on there, it might die because someone else has a priority job that's not a spot job. And so if you have a short running workflow like QuickMer runs in about 2 hours, then you can actually tolerate this pretty well. And we found that we could improve our cost from $1.43 a sample down to 25 cents. And I was like, yes, woo, we can make the budget go farther, which is always a good thing. It means we can analyze more genomes than we thought. And so we've analyzed about 32,000 genomes through this workflow. And I was mentioning to Mike that our biggest time crunch is like in getting the dbGaP approvals. So we're trying to get those as fast as we can. But we've actually run everything that we have dbGaP approval for, and we have some other things that are in the process right now. And then we developed an initial tool to genotype specific regions. So I said we want to look at specific non-coding regions, but we actually started with just genotyping genes. What is the average copy number of a gene, right? And we wrote this first in like Bash, and it was really slow. It took hours. And then I got like on this Julia kick because people at WashU love Julia because it's like one of the founders is there. It's for no other reason than that. And I got it down to 45 seconds. So Jack came into my lab and I said, okay, Jack, I've got a tool. It runs in 45 seconds and let me show you what it does. So we can, we prototyped, I prototyped it. This was early days. I prototyped it to show that it worked really well. So we know that this gene KCD13 is in a region of 16p11.2, which is important in autism. And these are all the people that had duplications. And these are all the people that had deletions. And like it got all of them perfectly. It also got small single exon deletions like this deletion in CHD2. And I'm like, this is good. I mean, there's a lot more data. I'm not showing it. I'm just showing a couple of plots. And I said, okay, Jack, there's the program. So make it faster. And he's like, cool. I only program in C++. This is going to be awesome. And I'm like, okay, let's see. So he's actually really amazing. And so he developed this tool called CMPI, and it can do genotype statistics of any regions you want, like all the genes in the genome. It can do digital karyotyping, right? So you can see like 46XY, 46XX, or Down syndrome. You can see it all. And it can do chromosomal sex determination based on that on the Y chromosome. And this is the workflow. So essentially, he has the QuickMer file, which I said it's generated for 25 cents, right, in about 2 hours in the cloud. And then we have a reference file, which is essentially some kind of an annotation file. And he's got some tricks to make it optimize here. And then he runs it into the program, CMPI, processes, gives all the information, and he has a plotting tool. And remarkably, I've tried it myself. He can do this. He can genotype all the genes in the genome in 1.75 seconds with one CPU and actually less than 1 GB of RAM. But he's like, well, you should just be conservative when you're talking about it, 1 GB of RAM. And I'm like, oh my God, this is amazing. So here's a couple of outputs from this. These are some of the regions and some of the information. So it will give you like the chromosome start, stop, the region, the average, the weighted average it's weighted based on the length of the region, the standard deviation, the coefficient of variation, the region size, the windows that came from the QuickMer file, and then the variation type. And he also has been able to integrate this plotting feature where this is an individual with trisomy 21, and you can see that there's 3 chromosome 21s here. Here's a person with autism who has 3 X chromosomes, so they're 48XXXY, and so there's the 3 X's there. And then the features of the tool can also do gene level plots. So these are the beginning of the windows that are seen in the QuickMer file, and then the little colored blocks are the genes. And this is obviously a duplication of several genes, and then he has a small region information output. So I was like, this is amazing. So now all we're limited by is getting access to the data. And the output of these files are very, very small. And so I think like if we have the QuickMer files from any resource like UK Biobank or Terra, like they're almost like trading cards that you could use for genotyping any regions of the genome, and now you can genotype them laser fast. So I was sitting there in the audience and I was thinking, I wonder if we could somehow get a tool like this into Terra, like that could just be run very quickly by anybody across those larger data sets, even if we don't have access. And of course, like we can put it up in the doc store and everything, but I was just trying to think like, what could we do? So the real reason though why I wanted to look at the genes first was because I was thinking it's a good place to start. And I added this slide at the last second. And so hopefully I don't regret that. But but what I was telling Jack was what I really want to do is I want to look at the copy number scores per individual, like a bulk aggregate score. And so I said, what I would like you to do is to actually take every gene and build out a distribution of copy number. And then we're going to score people in separate data. So we did that on 1,000 genomes. We threw away all the people who had crazy karyotypes from 1,000 genomes. We only used the ones that had good karyotypes, so the standard karyotypes. And then I said, let's actually make scores whether people sit on one end, like out past the 99th percentile for kind of like increased copy number, or less than the 1 percentile based on that distribution. And so we first generated the scores on HGDP, and I dropped the Y chromosome here because the Y chromosome skews the data a little bit. But we essentially made these scores. So if you have a deletion, we give you a score, or I should say, if you're at the extreme low copy number, we give you a score of 2 for that gene. And if you're at the extreme copy number on the high side, we give you a score of 1, because we tend to think deletions are more severe than duplications. You might ask, like, why would you do this if the copy number is all at 2? Well, actually, this analysis allows for us to look at those copy number genes where they're higher, right? Some genes in our genome are a copy number of 6 in most people, or 4. And so that allows for us to look at that. So he generates a score for each individual based on that information. And if you're in the middle part of the distribution, your score for the gene is 0. You're just like everybody else. And so what we did was we applied this to HGDP, and then we also applied it to 2 genomes from 9p deletion syndromes. So 9p deletion syndromes tend to have large contiguous gene deletions, and so we would expect them to have really high scores. And in fact, they do. So this is one individual and this is the other. We're still trying to work out this part of the distribution for HGDP, but by and large, the 9p are very high. And so I think what's kind of cool about this is that 9p is like the test case. It should look that way. But what about people out there who have copy number that's elevated in many parts of the genome, not just in a large contiguous block? And how can we find that? And so that's something we're working towards. Yeah, just a preview of where we're going. So in conclusion, for this part, we've been able to optimize existing tools to get really cheap, I think, and fast copy number estimates. We've been able to use a advanced, well, I shouldn't say advanced, but like a better programming language to increase the speed. So again, increasing the speed down to 1 second or so. And then we can do this for any regions of the genome, not just the genes. Can do it for anything. And I think that's really cool. So I thought I'd spend the last couple minutes talking about using the power of the cloud practically, because I wasn't sure of the audience. So I asked Dolly, right, to tell me. I said, imagine you're a researcher and you're working in the cloud and you have access to all this genomic data, and it's the future. And after a while, it gave me this, but I think the lady kind of is a droid, maybe. I don't know, because she's got like stuff going up her back. But and the DNA may not all be drawn correctly. I'm a human geneticist, so, but that's the best the AI could do. But I think it's pretty cool because, like, actually all of you are kind of like that when you log into your computer. You're like, you have access to everything just like that. And so some of the items to consider. So as a PI, I actually did some cloud computing during my postdoc, and I learned a lot during that process. So when I opened my lab and we got the AnVIL grant, I was like, okay, we got to go into this very systematically. And so one thing that we wanted to think about is, like, there are different pay points, and not that I don't want to make it all about money or anything. I'm just like pointing out some obvious things, like you store the data, you transfer the data to a storage unit where, attached to the compute node, and you have the compute node for the analysis. And I think what it's really cool is that you can think of it like renting a car. And I know a lot of geneticists use car analogies, but I couldn't get away from this one. I've gotten away from it in some of my comparison of sequencing technology, but I couldn't think of another today. But if you think of it like renting a car, maybe you just need a compute node that's super fast, like a Corvette. Maybe you don't. Maybe you just need a tiny car that's super efficient. Or maybe you're doing something with lots of RAM and you need to haul a lot of information. Like we have a 3 terabyte RAM node, for example, and sometimes you need it. It's kind of crazy. Hopefully not very often, but sometimes. So you might want to think about that. You want to think about bringing your data back to your cloud location and maybe bringing it to your local instance if that's something you want. Docker Hub fees, they're very small, but they're there. And then you want to think about who's going to run the cloud analysis and telling them, like, start really small and identify someone who's very responsible. So I have a person in my lab who's Jeff, who's very responsible, at least hopefully not running anything right now, but that makes me regret that. But but anyway, he's very responsible, and also realize that it's going to take some time. Like I talked to some PIs, and they're like, "Oh, my person didn't figure out how to run it in the cloud. It's been like 1 day," and I'm like, "Oh my God, give them a minute." Like it's a little bit something new for them possibly, and so it's okay. But once they get it, then it's really really easy. And then obviously to check in, and then what are the kind of workflows you might want to do in the cloud? And so I'll be honest, we don't do everything in the cloud. I mean, we just don't. I mean, but some things we have to. And one of those are instances where the data is really big. And I was talking about the running Quickmer too. Like we're never going to download 40,000 genomes to our server to run a program that can run for 25 cents in the cloud. It makes no sense for us to do that. So that's a very good instance of using the cloud. Or maybe you just don't have a lot of compute power. We have a lot of resources at WashU, but not everyone does. And so you might want to consider that. And then I think the cost modeling is really important. So, we do a lot of development on site. You learn how bad you are at optimizing your programs locally once you get into the cloud, because once you're in the cloud, you want to make it as cheap as possible so you can make those cloud credits or dollars go as far as they can. And so you can make them even better on site first. And then you can benchmark them and appreciate it might work better or worse in the cloud. Like it might not perform the same. And then determine if you can tolerate the spot VMs because they can save you a lot of money. But if it's not a workflow that's going to be able to tolerate those, it's not worth it, right? You might as well just run it the standard way. And then actually I asked Jeffrey to give me some slides on what it's like for him working in the cloud. And so these are some slides he put together. So first of all, perform local testing. So like he said, we do all of our first testing locally. And then, for example, he gave me this from Jellyfish, which was a program he was trying to run. And, looking for this good runtime RAM and input output. And so we had run it on different Kmer links. So it's a Kmer counter. And basically on Illumina data, PacBio data, and we found like kind of the optimal for Kmer length and memory. So we've got runtime, memory, output. And then just running it on one sample helps first, right? So these were kind of the data that he got from running on, actually it's Illumina and PacBio, so it's 2 samples, but it's the same person. And then scale up. So he usually does like 5 to 10, and then once 5 to 10 work, he comes tells me, he's like, I'm ready to blast it out to 10,000. And it's like, okay, we're good then. And so that's pretty important. And then another thing that he mentioned was like balancing the load. So if you have a whittle with lots of different steps, like balancing what each of those steps is doing in terms of time and usage. And then the input, sorry, I'll go back. And the input size and minimizing the tasks. So those are the, I'll stop there in terms of that. These are the resources, the computational tools, and I didn't list the ones that aren't published yet here, but and some data sets. So we do a lot of wet bench stuff in my lab too, and so we have data sets and protocols from that. And I will end with acknowledgements. The bulk of the work that I showed today was from Jeff and Jack, and like I said, they'll be around later this week. And I am recruiting postdocs on both the wet and or dry in case you're a hybrid. If anybody here is interested in kind of accelerating computation, be happy to talk with you. And I will end there and acknowledge my funders as well. Thank you.
Thanks for that great presentation. I think one thing I learned, it's always worth it to get the SSD. That's
Yeah.
like, anyway, but I was really excited about the opportunity for sharing some of what you're generating. I think you mentioned this, but I think one thing that we're interested in, I don't know if you've talked with NIH about this or thought about it, when you've done the quick Kmer, you've done the CMPI, you've generated those really important duplication scores. Is there a way to recontribute that? Have you thought about actually submitting those back to repositories? I know this is very complicated from a governance standpoint, but I'm just curious if you've tried to do it, if you've encountered problems, if you thought about it.
Yeah, okay. So for like de novo variant space, it was easy because we did 1,000 genomes and we just could make it available online. We actually made it on a Globus endpoint at that stage. And for the autism data sets, they can be contributed back to the Simons Foundation, which is like our main autism funders other than the NIH. But for these newer KMER-based copy number files, so this is like really new. We started getting this advanced CNPI in May. So like that's a really short time frame in science. We really want to share them, but Jack and I have been talking a lot about like, what can we do now if we can't? And so the thing we thought of with that scoring system, we're going to release the distribution scores for every gene. Like at least that part would be a way to start, so you would know if your person, so we have a supplementary script that can actually score your person to see like, are they outside of that? Ideally, I would love to contribute them all back. So all the KMER files, they're megabytes in size, so they're very small. And like I said, I think that they could be like trading cards, and people could genotype any region. Just because we care about genes or this one set of Crees doesn't mean everybody else, cis regulatory elements, sorry, I using shorthand there. But yeah, we'd love to do it. I don't know how to do it.
Sure. I think
Really.
it'd be great if you want to engage with us and we can talk about how you could do it, because I think it's so important to be able to share in open science
Yeah.
and be able to push that out there. Here's the
Yeah.
actual calls.
Yes.
Anyway, it's complicated governance wise, but would love to engage on
Yeah,
that.
it's a lot of different data sets too, and permissions. Like you were talking about the one of you were talking about the GRU versus the other health one that's not population based, HMB. Yeah, like those are a lot easier, but then some of them are like autism and they're disease specific, so it gets trickier there. But we definitely want to share, especially because I think that helps with reproducibility, not just like using it for new things, but people can check what we did too. I always think that's important.
Thank you again. And you were mentioning looking for data. I think when you're talking about the trios, is there sort of like what's the next mountain in front of you in terms of data you're looking for? Do you wish it, do you know what you want? Does it not exist? It exists and it's hard to get to. How would you think about that?
I think like the easy thing to do is, so we just got a grant from Kids First too. So we're just getting into that space as well. And well, one thing I mentioned at dinner last night was that for Kids First, we were highly encouraged to use Cavatica. And we're like, okay, it's a new system and that's cool. But we also looked at Kids First data and it's available to us. I mean, it's not available to us. It's in Terra. But on Cavatica, we can access it even though we have like DB gap approval, but in Terra, we can't access it even though we have DB gap approval. And I don't know if like I checked the wrong box and only did Cavatica. But so I think for me, it's like, how do we get the genomes easily so we can run our tools? And I mean, of course, we want to be responsible because that data comes from real people who consented and everything. I think that's the challenging part. Like we're accelerating the tools, but then getting access to the data is the next level. It's really easy to get GRU, HMB, but then the disease specific, like for Kids First, for example, it's like, I don't know, 12 different disease specific. And so it's like going back and resubmitting and it's just paperwork. It's fine. But I wish it were easier. That's all I'm saying. Yeah.
So I think I have two questions. So one, if I recall, Simon Simplex collection was like a mix of saliva and blood and other tissues. Is there any concern about the de novo rate when you look at mixed tissues?
So Simon's is all blood and Spark is all saliva. So there are two separate autism cohorts, and the de novo rates are very similar in both. And so we think it's just because they're not being cultured at all.
That's the key.
Yeah, Simon's does have LCLs, but they don't do any sequencing on LCLs. They just have them in case people want to do something else with them down the road, like confirmation of de novo variants, because like or other kinds of variants, because you don't need like, if you just want to make sure it was really there, you can use any DNA.
Okay,
Yeah.
and then I think my last question is so thank you. You have our full attention.
Oh, okay.
If there's anything that you could ask of us, what would it be? Like, what's your biggest pain point? What's your biggest need? How can we help?
The biggest pain point. Well, what I would like to see is like, is there a way that this sounds weird, but like that Terra could actually integrate biobank data from other countries, like UK Biobank? That would be super cool. Like one home for the data. I don't know. That would be my dream.
I
All the
love it, though.
data,
I mean, it's
one place.
interoperability, right? How can we
Yeah.
get access to all these different data sets?
Yeah, because that's like a whole nother level of regulation and would be super cool.
I agree.
Yeah.
All right. Thank you so much.
Thank you.